pituitary. We'll talk about the posterior pituitary first because it's a little bit easier to remember. There are only two hormones that get released from the posterior pituitary, oxytocin and vasopressin, also known as ADH, antidiuretic hormone. The thing about the posterior pituitary, it's like an extension of the hypothalamus, okay? No hormones actually get made in the posterior pituitary. They just get released from the posterior pituitary. Oxytocin and vasopressin both get made in neural secretory cells of the hypothalamus, and then they get released from the hypothalamus into the posterior pituitary, where they then get released from the posterior pituitary to travel to their destination and their target organs. Oxytocin's target organ is usually smooth muscle contractors. The target organ for vasopressin is what? You'll learn about it in a little bit.
Vessels?
The kidney, okay? The collecting duct specifically, okay? So that's the posterior pituitary. Then you have the anterior pituitary, okay, which is connected to the hypothalamus through these portal vessels, okay? These portal vessels are super important. Why? What is the function of the portal vessel? It's direct delivery of whatever hormones are getting released from the hypothalamus into the anterior pituitary, so that these hormones don't have to travel and circulate any farther than just going straight to the anterior pituitary. Okay? So portal vessels are super important for direct delivery of these hormones from the hypothalamus to the anterior pituitary. The anterior pituitary works a little bit differently in terms of it actually makes its own hormones depending from stimuli from the hypothalamus, okay? So the hypothalamus gets its signal, it makes a certain hormone, it releases this hormone into the anterior pituitary and signals the anterior pituitary to either make an inhibitory or activating hormone that will get released into the endocrine system or into the blood. Your book gives you a list of these hormones that get released from the anterior pituitary. I would focus on the ones responsible for the thyroid, obviously oxytocin and vasopressin, and prolactin, because Dr. Purcell went over those in class. Yeah. Here's just my slide on portal vessels. We already went over this. Again, it provides a direct route to the anterior pituitary without circulating the rest of the blood to get there. Another concept to understand is that the pituitary gland is not only releasing hormones that are going to be activating certain target tissues, but they also could be releasing hormones that are going to negatively regulate certain things as well. This is the simplified pathway of thyroid hormones that get released from the hypothalamus and anterior pituitary, and we'll go into a little bit more detail of that with them in a second. But like I said, here we have the stimulus that's being received by the hypothalamus. It calculates and sees what output it's going to put into the anterior pituitary. In this case, we need to increase our metabolism. So the hypothalamus is going to release TRH, thyroid releasing hormone, into the anterior pituitary via those portal vessels. Once the TRH gets to the anterior pituitary, it's going to release TSH, thyroid stimulating hormone, okay? And from here, the thyroid stimulating hormone is going to travel to the blood, to the thyroid, and stimulate the production of what? T4 and T3, okay? We're going to talk about the specific mechanisms of that in a second. Let's just talk about prolactin for a second. Prolactin can get released from the anterior pituitary and is responsible for milk synthesis. It has some interesting things going on. So milk synthesis is necessary after childbirth in order to feed the child. At this point, it would make sense if the mother is focused on feeding her current child that prolactin has some inhibitory mechanism on the menstrual cycle to prevent another pregnancy for some given amount of time. Okay? What else thyroid hormone. This makes sense. This is just, a regulatory mechanism to regulate the release of prolactin from the anterior pituitary. We already talked about oxytocin, but just to show you the slide on it, oxytocin is released from where? Posterior pituitary. It is made where?
Hypothalamus.
Hypothalamus and the neurosecretory cells. It gets released and it goes to its target tissue, which is what in this case? The uterus, right? And it's promoting these contractions that we talked about earlier in order to promote childbirth. All right, so now let's get into the specifics of the thyroid gland. We said the thyroid gland makes T3 and T4. And we're going to talk about specifically how it does this. But the thyroid gland is responsible for regulating metabolism. T3 and T4 are going to be responsible for regulating metabolism through signal transduction pathways that are going to express genes responsible for metabolic processes. Sorry, and also I said thyroid releasing hormone is actually thyrotropin releasing hormone, just in case, but you can just know it as TRH. We went over this pathway already, but I'll go over it again. The brain receives a signal, the hypothalamus receives a signal. It decides to release TRH into the anterior pituitary via the portal vessels. The anterior pituitary is then stimulated to make TSH. TSH gets released into the blood and travels to the thyroid, where it's going to activate its mechanism to make T3 and T4. So this is what cells of the thyroid look like. These are follicle cells that are sort of on the outside, and they create this lumen, okay? Usually this is what follicle cells look like. When you learn about reproduction and the ovaries have follicle cells as well. Dr. Brasal pointed out that tissues with follicles are most likely to get cysts, cystic ovaries, cysts on the thyroid. This is, I guess these cells have a predisposition for these kinds of things dependent on their structure. We're going to be concerned with the follicle cells for the production of T3 and T4 and a little bit of the lumen. Before we get to that, let's talk about the structure of T3 and T4. T3 and T4 are made from tyrosine. You can see based on the structure of tyrosine that, do you think it's going to be, the T3 and T4 is going to be easily transported in the blood? No, because of those ring structures, right? So because it's poorly soluble in the blood, the T3 and T4 are going to be carried by thyroid binding globulin. Do not confuse this with thyroglobulin, okay? So thyroid binding globulin carries T3 and T4 throughout the blood into the various cells it needs to in order to regulate metabolism. The difference between T3 and T4 is easily seen. T4 has 4 iodines, T3 has 3. Based on this structure, they're going to do different things. T3 is the activated form because it has 3. More specifically why, I don't really know. That would be something you could look up. But because it has 3, that's its active form, and it can go on to bind with whatever transcription factors it needs to express genes for metabolism. So let's talk about the production of T3, well T4, really. So T4 gets made on this RAF protein called thyroglobulin, where it takes the tyrosine and thyroid peroxidase works and adds the iodines and puts the 2 ring structures together. Don't confuse these. Thyroglobulin is essentially this RAF protein that is holding the tyrosine and assisting in putting together the structure, and thyroid peroxidase is the one that's adding these iodines onto the T4. Alright, so where is this all taking place? So you see these follicle cells that we just talked about in the other slide. Here's the lumen, or the space of the thyroid cells. that iodine is necessary in our diet in order to make these molecules, okay? Without it, we could see some problems that happen due to lack of iodine. So iodine needs to be taken into the lumen of the thyroid cells, where thyroglobulin is then going to be released from the follicle cells. From there, what do we say thyroglobulin does? It doesn't carry, it's sort of like that raft protein that's assisting in the production of T4 and T3, right? So then, the thyroglobulin and the thyroid peroxidase make T3 and T4, and then there's some hydrolysis that goes on. Why? Because you have to release the T3 and T4 from the thyroglobulin, right? So they were on, like I said, they were on this raft protein, and then they have to be released into the blood, so there's some hydrolysis that has to go on. And then T3 and T4 are released into the bloodstream to go act on cells responsible for metabolism. Any questions on this mechanism? On thyroglobulin? On thyroid binding globulin? Yep.
What is endocytosis?
What is endocytosis? You tell me. You guys learned about that last semester. One person. Definition. You guys should know this. Yeah. Yeah, the cell sort of like forms a little membrane around whatever molecules that need to either come into the cell or exit the cell. Okay? And then there are these clathrin binding proteins that assist in this. This is last semester. Good? Any other questions? Yep.
So the thyroid binding globulin carries
in the bloodstream. So once the T4 and T3 are released from the follicle cell, they need to be assisted in transported through the blood because of their structure. So the thyroid binding globulin is the one that carries T3 and T4.
The thyroid globulin is the one that assists in
the production. Yeah. All right, last slide. T3 and T4 are carried in the bloodstream to the cells, their target cells, that they need to activate gene expression for metabolism, whatever that pathway will be. T4 needs to get turned into T3. It does so with the help of this D1, D2 complex. How do we turn T4 and T3? What was the difference? The iodine. You take that iodine off, right? That's essentially what's happening. From here, T3 is now active. It's able to enter the nucleus. Again, think about these regulatory mechanisms, right? Maybe the reason why we regulate this is because the nucleus only recognizes T3 in its nuclear pore to transport it inside to activate this gene expression. These are like little things you could think about, right? So the T3 is able to enter the nucleus, and it binds this THR protein. It makes this transcription factor complex that we talked about. This transcription factor complex that we learned about last semester. What does it ensure? Binding of what? RNA polymerase, right? So it creates this big complex to ensure specificity and call RNA polymerase over to express this gene, okay? Dr. Purcell gave you in this slide one example that, could be activated is the uncoupling protein, the UCP protein that we talked about in the uncoupling process for the non-shivering thermogenesis and other metabolic processes. Any questions? All right, guys. Good luck. Justin is here.